You know, in the world of medicine, the next huge discovery could be hiding right under our noses. Today, we're diving into how AI is being used to hunt for new drugs in, of all places, the fungal kingdom. It's a quest that honestly sounds like something straight out of science fiction, but as we're about to see, the reality is, well, it's a lot more complicated.
So why all the excitement? It really comes down to this one number, over 99%. That's how much of the chemical world produced by fungi remains completely, totally unknown to science. We have barely scratched the surface of what could be a massive, mysterious world of potential medicines.
And that leads to this huge question, right? We all know that the most famous antibiotic in history, penicillin, came from a simple, common mold. So what else is lurking in that massive, undiscovered chemical library? Could the cure for the next superbug or some revolutionary new drug be waiting for us in there?
And look, this isn't just a theory. Fungi are a proven source of medicine. We rely on them for absolute game changers like penicillin, for the statins that millions of people use to manage cholesterol, and for crucial immunosuppressants that literally make organ transplants possible. This track record is exactly why scientists are so hyped about what else we might find.
But here's the catch. There's a massive bottleneck. The way we've traditionally discovered drugs is like trying to navigate a maze in the dark. It's an incredibly slow, ridiculously expensive grind. We're talking years and millions of dollars, which makes exploring that vast 99% a truly monumental challenge.
So, to navigate this maze, you need a better map. And this is exactly where AI enters the picture, promising nothing short of a revolution in how we search for these hidden cures.
Okay, so let's get into a really key concept here, the biosynthetic gene cluster, or BGC for short. The easiest way to think of a BGC is like a molecular recipe card that's written directly into an organism's DNA. It's this group of genes that all work together to build one specific chemical compound. So finding these BGCs, that's the critical first step to finding new drugs.
Now, in theory, the AI process is just revolutionary. Step 1, you sequence a fungus's genome. Step 2, an AI model scans all that DNA and finds the recipes, the BGCs. Then, and this is the magic part, the AI actually predicts what those recipes make. Is it an antibacterial, an anti-tumor agent? This would let scientists jump straight to testing the most promising stuff, saving an unbelievable amount of time and money.
But that's the dream, right? How well does this amazing idea actually work in reality? Well, a brand new study from 2024 decided to put this exact idea to the test.
So let's set the stage a little bit. Researchers had already built AI models to do this for bacteria, and they were really successful, hitting up to 80% accuracy at predicting what a compound does just from its BGC. So the expectations were pretty high. The team took these proven models and adapted them for the much more complex world of fungi, really hoping for similar results.
But you know how it is with science. It rarely follows a straight line. What they found wasn't the big breakthrough they were hoping for. Nope, it was a major plot twist. And the results were stark. The AI's accuracy in predicting if a compound was antibacterial was only 58%. For antifungals, a little better, 68%. And for anti-tumor properties, just 61%. I mean, these numbers are a far, far cry from that 80% success they saw with bacteria.
Let's put it bluntly, these models performed barely better than random chance. A 50% accuracy is literally what you'd get from just flipping a coin. So what on earth is going on here?
This is the million-dollar question, isn't it? If these powerful AI algorithms work so well on bacteria, why did they just fall flat when they were faced with fungi?
Well, the answer, as it turns out, has very little to do with the AI itself. The problem wasn't the algorithm. It was the information we fed it. It's a classic data problem.
First, let's just take a moment to appreciate the sheer scale of this. Scientists estimate there are literally millions of these BGCs, these chemical recipes, tucked away in the genomes of all the fungi on Earth. It's an immense, incredibly diverse library of possibilities.
Now for the reality check. The public database that was used to train this powerful AI contained a grand total of 314 fungal BGCs with known functions. Not millions, not even thousands, just 314.
This analogy just brings it all home perfectly.
Imagine trying to build an AI that can identify any animal on the planet, but your entire training library is just a few dozen photos of pets from your street. The AI wouldn't fail because it's dumb, it would fail because it has absolutely no idea what an elephant, a whale, or a poison dart frog even looks like.
And if you can believe it, the problem gets even worse. Not only was the data set tiny, it was also super biased. Almost a third of the data came from just one single genus, Aspergillus. If you add in Penicillium and Fusarium, just 3 types of fungi account for half of all the data. So it's not just a small library, it's a library that's almost entirely focused on just a few authors.
So this really isn't a story about the failure of AI. It's a story about the desperate need for a better map. The real challenge here isn't computational, it's foundational, it's about biology.
And here it is, the core takeaway from all of this. The thing holding us back isn't our AI technology, it's our biological knowledge. Before we can ask an AI to find the needle in the haystack, we first have to do the hard, fundamental work of actually building the haystack.
The authors of the study really said it best themselves. The urgent need right now isn't for a better algorithm. It's for systematic, foundational research that connects a fungal compound to its genetic recipe and then documents what it does. We simply need more data points on the map.
And this leaves us with a final, really crucial thought. The technology is here. The artificial intelligence is powerful, it's ready, and it's waiting to start the search. The real question is for us. Are we willing to do the fundamental, painstaking scientific work required to draw the map it needs to explore? The next penicillin might just depend on it.